Hello everyone, today I'll be presenting on African sleeping sickness, also known as human African trypanosomiasis, from a microbiological standpoint. This disease is caused by a fascinating but deadly parasite in the genus, Trypanosomoma. It's a perfect example of a pathogen that uses complex survival strategies to evade the human immune system. Understanding its biology is crucial for developing prevention and treatments. African sleeping sickness is restricted to the sub-Saharan Africa and is transmitted by the tsetse fly, which is of the Glossina species. There are two main forms of the disease, each caused by a different subspecies. Trypanosomia brucei is responsible for the chronic form found mostly in West and Central Africa. Trypanosomia brucei rhodescens is responsible for the acute form found in the East and Southern Africa. TB gambiense accounts for about 97% of cases and progresses slowly over months or years. TB rhodescens is a much more aggressive form and can be fatal within weeks. The causative agent Trypanosomia brucei is a flagellated protozoan parasite. Microscopically, it is a unicellular eukaryote with a long, slender body and a single flagellum attached via an undulating membrane. Key microbiological factors include kenotoplast, a unique DNA-containing organelle found in the kenotoplastids. It is part of a single large mitochondrion of the parasite. Surface coat, which is made entirely of variant surface glycoprotein, or VSG. Flagellum, essential for motility in both the insect vector and the human host. It has a complex life cycle which alternates between the tsetse fly vector and the Manna-Millennium host. Life cycle. In the tsetse fly, trypanosomes exist as procyclic trypomastogotes in the midgut. They migrate to the salivary glands, maturing into epimastogotes, and finally into the metacyclic trypanosomogotes, the infective stage. When the tsetse fly bites a human, the metacyclic trypanosomogotes enter the bloodstream. In humans, they transform into bloodstream trypanosomogotes, which multiply by binary fusion. The parasites circle in the blood and lymph and eventually invade the central nervous system in the later stages of the disease. Immune evasion. One of the most studied aspects of T. brucei in microbiology is antigenic variation, the ability to change its surface coat, VSG, regularly. Why does this matter? The human immune system detects parasites by their surface antigens. Every 7 to 14 days, T. brucei switches VSG expression in a different variant gene. This means the immune system is constantly playing catch-up, producing antibodies against one coat while the parasite switches to another. This antigenic variation results in waves of parastemia, cycles where the symptoms worsen as the parasite population surges, then temporarily subsides before the next surge. Pathogens and effects on the body. African sleeping sickness progresses in two stages. Stage 1 is the hemolymphatic phase. Parasites multiply in the blood and lymph. Symptoms include intermittent fever, headaches, muscle and joint aches. Swollen lymph nodes are also a common symptom. Immune response triggers inflammation, but antigenic variation makes clearing the parasite impossible. Stage 2 is the neurological phase. Parasites cross the blood-brain barrier, entering the central nervous system. This causes progressive neurological damage, confusion, sensory disturbances, poor coordination. The hallmark symptom is disruption of the sleep cycle, leading to daytime sleepiness and nighttime insomnia, hence the name sleeping sickness. Eventually, without treatment, severe neurological decline occurs, leading to coma and death. Diagnosis and laboratory work. Diagnosis often involves finding trypanosomogotes in the blood smears in the early stage, or cerebrospinal fluid in the late stage. Other microbiological methods include card agglutination test for trypanosomiasis, or CATT. For TB gambiense, molecular methods like PCR for parasite DNA. Serology detects antibodies. Treatment and challenges. Treatment depends on the stage. Stage 1, pentadimen for TB gambians, or sermon for TB rhodescens. Stage 2, meloropasol, is a highly toxic arsenic-based drugs, or euphenethrin for TB gambians. Newer combinations like NECT reduce toxicity risk. Challenges. Drugs for CNS stages are often difficult to deliver and have severe side effects. No vaccine exists due to the antigenic variation. Rural, low-resource settings make vector control and diagnosis very difficult. Prevention and research outlook. Prevention focuses on reducing tsetse fly exposure, such as insect traps, repellents, or clearing vegetation. Active screening in at-risk populations is also important. Research continues on finding ways to block antigenic variation, developing drugs with better CNS penetration and fewer side effects, understanding the parasite's genome to identify new therapeutic targets. In conclusion, African sleeping sickness is as much of a problem of microbiology as it is of public health. The parasite T. brucei is a master of immune evasion, and its ability to invade the central nervous system makes it both deadly and challenging to treat. Continued research into this unique biology offers hope for better diagnostics, treatments, and possibly prevention in the future. I'd like to apologize for any mistakes I made with pronunciation. Thank you for listening.